Assalamualaikum warahmatullahi wabarakatuh and hello everyone. Today, we from Group 8 want to present about inductive coupled plasma mass spectrometry or known as ICP-MS. Our group consists of 4 members which is Adam, Haikal, Effendi and Akmal. This is the thing that we're going to discuss about ICP-MS. Firstly, we need to know what is ICP-MS. ICP-MS is a type of mass spectrometry that uses an inductive coupled plasma or ICP to ionize the sample for analyzing process. ICP-MS also is a very powerful tool for analyzing trace metal in environmental sample. ICP-MS is being used to quantitatively measure most of the elements inside the periodic table. This is the list of the elements that can be measured using ICP-MS. ICP-MS is used widely in environmental, food, and industrial sectors.
So basically there is 6 components of ICP-MS. The first one is sample introduction, deliver finely divided sample to plasma. The second one is plasma generation such as ion source and AR plasma. The third one is interface, allow transfer of atmospheric pressure ion source to a high vacuum mass analyzer. The next one is ion optic, focus ion beam and help eliminate neutral species and photon. The next one is mass analyzer, spread and measure individual ion by mass. The last one is vacuum system, provide low pressure environment for mass spectrometer to operate. Sample introduction. For sample introduction system, the first one is provide the mean of getting sample into instrument. The second one is compose nebulizer and spray chamber. The third one is the liquid sample introduced to nebulizer by peristatic pump through self-asphyxiation that create an aerosol of fine droplet. The last one is the fine droplet are passed through a spray chamber before they are allowed to enter the plasma.
The second mechanism in ICP-MS is the torch. It consists of radio frequency or RF coil that connected to RF power supply. The torch in ICP-MS is used to atomize and ionize sample for interference. Startup spark from Tesla unit will be applied and argon gas will enter RF field and form plasma. So, what is plasma? Plasma is an electrical conducting gas mixture. It contains sufficient amount of cation and electron to maintain the conductance. This is the process that occurs in the torch. Firstly, nebulization. It will convert liquid sample into aerosol for introduction into the plasma. Then dissolution will occur, where the aerosol solvent is evaporated and leaving only the analyte. The analyte then will be undergo vaporization, where the analyte is vaporized into gas. The high temperature, around 6000 Kelvin, inside the torch will complete the atomization process. And finally, the atom is finally ionized via ionization process.
Next is ICP-MS interface. During the process, the sample ions pass through the plasma vacuum interface region situated between two cones. Two types of cone involved are sampling cone and skimmer cone. Sampling cone only allows a fraction of the analyte and argon ions to pass through, while skimmer cone to reduce the numbers of ions allowed into the mass spectrometer. Pressure reduction in the interface region causes the incoming ion beam to dramatically expand behind the sampling cone. The orifice of the second skimmer cone consistently passes on an ion distribution that is representative of the central analyte portion of the plasma. This process maintained with a mechanical roving pump. The interface vacuum also serves to remove most argon atoms at this point. Why we need vacuum system are to remove all the gas molecules in the space between the interface and the detector, and to avoid collision between ions and gas molecules during travel. In the system, we will use two roughing pump and two turbo pump, which are one large and one small. Large turbo pump is used to maintain low pressure just behind the interface, while for the small, to maintain the very low pressure at the mass analyzer and detector. For roughing pump is to create a vacuum system for the mass spectrometer.
Hello everyone, my name is Akmal and now I'm going to explain to you about mass spectrometry. You need to know the basic principles behind mass spectrometry. What we have here is mass spectrometer. It separates the singly charged ions from each other by mass. In other words, it is a mass filter. It has quadrupole, which consists of 4 parallel rods arranged around a central axis as shown below. Voltage and radio frequencies are applied to the rods, with one pair of rods having opposite polarity to the other. By setting the RF voltage to only ion of selected mass to charge radial passes, the stability within the rods can be maintained. Afterward, the selected ions will be transmitted toward the detector, while the other ions are ejected and collide back to the rods. Next, we have the detector. You can refer to the picture and diagram provided to help you visualize it better. After the selected ion passes toward the detector will amplify electron through the dynads contained in it. You can refer to the picture on the right. As the electron passed the first dynads onto another and another, the number of electrons grew. That means the number of electrons increases as it strikes more dynads, therefore making the pulse easier to measure when it grows bigger. Then the signal received will be digitized to make it easier to read.